So these examples of the bacterial artificial chromosome and yeast artificial chromosome. So here the Yak cloning in yeast, BAC means bacterial artificial chromosome, and P1 derived artificial chromosomes in bacteria, and mammalian artificial chromosome and human artificial chromosome. Okay, so in mammals and human cells these are derived, and here just we have a bacterial and yeast artificial chromosomes. So this bacterial artificial chromosome or BAC are the plasmid designed for the cloning of a very long segment of DNA. So here first BAC vector, that is PBAC 108L. So cloning of a large regions of the eukaryotic genomes and origin of replication from the bacterium E. coli, F factor, and BAC vector as per the PBAC E3 6 and PBE LOBA C11. Okay, so these are the examples of bacterial artificial chromosomes. Okay, so here this gene produced enzyme beta galactosidase as well as stable origin of replication that maintain plasmid. So after recombination, the circular plasmid BAC containing large target DNA. Okay, so DNA plasmid DNA plus inserted DNA is plasmid BAC containing large target. Okay, so DNA inserted to the host bacteria by the electrophoresis. So here this is used in analysis of genomes. Okay, so genomes analysis means which gene can control which type of characters, so etc. The host of BAC is mutant strain. Okay, so mutant strains, mutations occurs are done in that chromosomes or genes. So this is about bacterial artificial chromosome. And next, yeast artificial chromosome. So YAC can accept 500 to 3000 kilobytes of DNA or kbs of DNA. Due to their greater capacity, these have replaced former ones. So YAC contain all elements needed to maintain a eukaryotic chromosomes in yeast nucleus. So a yeast origin of replication, selectable markers and sequence needed for the stability and proper segregation of chromosomes at all divisions. So liner, it is this vector is also a liner plasmid vector. So cloning large DNA segments 100 to 1400 kbps, occurring two forms: circular means grown in bacteria, liner multiple in yeast cells. And here the example PYAC3. So first YAC developed means yeast artificial chromosomes was developed. One is PYAC, and next it contains ARS sequence means replication sequence, and CEN4 sequence centromic functions, and TRP1 and URA3 two selectable markers. So here use mapping complex eukaryotic chromosomes means eukaryotic chromosomes, it is useful for map to form the or arrange the mapping of eukaryotic chromosomes. So it is useful for chromosome mapping, eukaryotic chromosome mapping. Then next, here the applications of vectors. So a particular gene can be isolated and it is nucleated, sequences determined, and control sequences of DNA can be identified and analyzed. So first, in what we have to do, just isolate what is that, the desirable gene. A particular gene, that is desirable gene means the gene having a good characters means nothing but for suppose it have a resistance for some what is that, the bacteriums, viruses, or it can produce a number of fruits, or it can produce sweet fruits, whatever it is. Okay, so it control a such type of character. So that particular desirable gene we isolated and its nucleotide sequence determined. First we determine the sequence of that DNA, and next control sequence of DNA can be identified and analyzed. So then the sequence can be identified and analyzed then after. So with the help of these back vectors, And next, protein or enzyme or RNA function can be investigated. So with the help of these vectors, the protein, enzyme or RNA functions also investigated, and mutations can be identified. For suppose unfortunately some mutations occurs in means either gene mutations and chromosomal mutations, particular the gene mutations can identified with the help of vectors. Example, gene defects related to specific diseases, gene defects related to specific what is that diseases. So that's why this is useful for identification of mutations. Organisms can be engineered for specific purpose. Okay, so these for suppose genetically modified organisms or genetically engineered organisms can useful in specific purposes like what is that we produce what is that uh, in the what is that gaining of milk and good quality wool. Okay, so and meat. Okay, productivity, increase the productivity of milk, eggs, wool, meat, whatever it is. Okay, so that type of particular animals. Okay, so that particular gene can collected and recombinated and entering into the animals, so that's why the productivity was improved. So this is also a what is it, a definite or a particular application of these vectors. Vectors are used in the DNA technology or the cloning, animal cloning or plant cloning. Okay, so just this is about the applications of cloning.